Welcome to everybody. I'm Claudio Doglioni, Professor of Pathology at the University San Raffaele Vita-Salute in Milan. And it is a pleasure for me to introduce Caterina Eloy. Caterina Eloy is a professor of pathology at the Faculty of Medicine of the University of Porto, in Portugal, of course. And she is the head of pathology laboratory at the IPATIMUP. IPATIMUP is the acronym of Institute of Pathology and Molecular Immunology of the University of Porto. It is a research and clinical institute founded in 1989. Sobrino Shimoes is a well-known pathologist, is in the board of directors of this institute. And the mission of IPATIMUP is research and innovation in health, and the career of Professor Caterina Eloy perfectly represents this mission. She is young, but a very brilliant curriculum, with her main research and clinical interests focused on thyroid pathology, cytopathology, and digital pathology. She is the author of several relevant scientific papers dealing with the application of digital pathology and artificial intelligence in cytopathology. She is an active member of several pathology societies, among them in particular the European Society of Digital and Integrative Pathology, of which she was president till 2022. Innovation is not only considering advanced technologies, but means also to improve lab organization and workflow, and the activity of Professor Caterina Eloy is also focused on this. It is important to maintain our attention on the basic fundamentals of our activities, starting from the beginning of the tissue and cell sample preparation, and how this aspect could impact on the final results. Today, Professor Caterina Eloy will speak about how some of the initial steps of tissue preparation can interfere with some of the diagnostic tests we routinely perform, so the PD-L1 evaluation. Thank you, Caterina. Now I let you your presentation.
Well, dear Professor Doglioni, thank you so much for this beautiful presentation. I do thank you very much, dear Pat, for inviting me to be here today. And I will show my screen. Please, it's everything good? You can see it well, right? So what we will talk about today, it's about how tissue processing per se may interfere in the way we evaluate biomarkers. And I will use PD-L1 as an example, since this is a widespread used biomarker, and we know how sensitive it may be to all pre-analytic and analytic conditions as well. So we are all aiming to introduce precision in the way we approach our specimens, the added value of pathology in terms of reporting and integrated information in terms of prognostic value and predictive value of response to therapy has been stressed, and it's tremendously increasing in the last years. So in a certain way, we should all pay attention and fight for the best determinations, the most precise determinations of biomarkers. And PD-L1 is one of these examples. We know that PD-L1 is one of the elements of these immune checkpoints. It has been appointed as a very relevant biomarker to determine the expression, to determine the response to immune therapy, therapies that are very expensive, that have side effects. And we know that although it has been demonstrated to carry important information, it's a complex biomarker that also carries some sort of fragilities. I just brought this brought these tables from the beginnings of the implementations of PD-L1 testing. I mean, PD-L1, it's there for almost 10 years, so it's not something new. This was and still is our issue from the beginning. We have companion diagnostic antibodies that are related to each type of treatment, and this means a multitude of approaches, multiple antibodies, multiple detection systems, including instruments. So the package that we need to use for a certain determination for a response to therapy can vary a lot. At a certain point, and this was starting with the non-small cell lung cancer, at a certain point we realized there are some possibilities of using these antibodies in an interchangeable way, so to use one to evaluate the response to all, more or less. This is not transversal. There are exceptions, but there are some that are very, very similar in one hand. So these differences have been attenuating as time passes by. On the other hand, new cancer models are being targeted. So we are now having immunotherapy used in breast cancer, in cervix cancer, in colon cancer, in gastric cancer, and many models now are being enrolled in the determination of PD-L1. So what we do in our lab, as this constitutes a major concern for us to have a very good determination of the biomarkers, is that we have a permanent control of all immunohistochemical slides that is supported by a complex tissue bank that we use to produce tissue microarrays that we use in every single slide. So all our immunohistochemical slides, all of them, and due to these specificities, PD-L1, it's really special. We introduce a complex of TMAs per slide with positive and negative controls. Each TMA has a map. We can track the origin of the tissue, and there is records, and we have a production that is semi-automatic in our lab. This is laborious, of course, but with this we have in each slide a tonsil, we have a leiomyoma, a placenta, and 2 lungs, one positive and one negative, that allow us to have an overview on the expression. And since we are working in a digital environment for more than 4 years now, we need to have this on slide so that we can even control the influence of scanning and screens in the intensity of the expression of these biomarkers. Why are we so concerned, double concerned, triple concerned about biomarkers at this point? Definitely quantifications that are more and more precise are in our goals. We have a lot of expectations about the use of computational softwares, mainly those that are AI, artificial intelligence based, and these softwares have the promise to start counting these biomarkers in a more accurate way. The fact is that artificial intelligence is also more sensitive than we are. We are very tolerant. We are tolerant to artifacts. We are tolerant to biased things that happened in the slide, but AI is not, and this constitutes a huge issue. It has been demonstrated already that deep learning-based softwares are incredibly biased by things like folds, bubbles, precipitates, and occasional contaminations within the tissue. This will lead us to incredibly wrong results. And the presence of these accidents in our slides is not so rare. We know it's frequent, and depending on the lab, can be tremendously high. So we can, as a person, as a human, we can ignore this interference, but artificial intelligence will not. And instead of doing a better quantification exercise, we'll do a bias quantification if we are not providing the best quality whole slide image for evaluation. It has been already demonstrated, for instance, in the setting of cytology, that if we change the type of processing of the cells, we will have distinct results in the classification by artificial intelligence, and this should worry us deeply. When we are reviewing the factors that are affecting the evaluation of biomarkers, either at the immunohistochemical level, either at the immunohistochemistry or molecular, we know that there are many factors. Some of them have been already studied, some are very well established, mainly for some groups of tumors such as breast cancer or colon cancer, but others have been completely under evaluated. They have not been explored, such as the type of reagents that are used in processing, the type of heating, if there is movement of the specimens or not, the manual versus the automatic processing, the clearing reagent and conditions, the dehydration reagent. So there are many steps in processing that are different from each type of instrument, and they will have certainly different impacts in the antigen exposure of the tissues. So, because we are doing this digital transformation and we are aiming to use artificial intelligence for quantification, we decided to do this testing in our lab. So we decided to see how could tissue processing interfere. Because if you follow my thinking, we can immediately see if there is a fold there, or if there's some cutting artifact there, or if there is an even cutting, we can see with our eyes. If there is an interference of processing, we just can't. So the amount of antigen that is exposed at a certain moment in a tissue, it's not possible to be evaluated by the human eye immediately. So we decided to test. And I would invite you to go along with me in this apparently very complex scheme, but you will see it's quite easy to understand. If you understand this, you will understand the design of the study that we made, and that you can probably reproduce in our lab as well to see how intense it may be the influence of tissue processing. So we tested samples that we know that are used as controls for PD-L1. We used leiomyoma, placenta, and tonsil. And for each of these specimens, which were many, we collected 3 fragments for each of the specimens. And each of these fragments, 3 fragments per specimen, they were inked with 3 different colors. So the red, the black, and the green. And then when time of processing come, we just gathered all the reds and we processed them. We gather all the blacks and we processed them. And we gather all the greens. And red, black, and green were processed in different processors. But then, when processing was over, we just put these fragments together with their sister fragments of the same specimen within the same cassette. So you see on your right a paraffin block with 3 fragments from the same specimen processed by 3 different processors. So with this analytics, we were just overcoming the interference of pre-analytical conditions, cutting conditions, macroscopy conditions. We were just introducing the variability that is inherent to processing. And then what we did with these paraffin blocks, these special paraffin blocks, we were cutting these paraffin blocks, and we were extracting H&D, PD-L1 22C3, and PD-L1 SP142 that we know are very different antibodies. We know that 22C3 may have some similarities with SP263, but not so much with SP142. We know they have different dynamics, so we choose to test them both. So for each paraffin block, 3 tests, 2 PD-L1 and 1 H&D. And then we entertain ourselves with this material. Slides were scanned with our routine protocols for observation in the P1000 from 3D Stack, and these whole slide images were reviewed by 2 pathologists. So regarding H&D, what were we observing? We were observing if there were artifacts related with processing and if there was an overall adequacy for diagnosis. As for PD-L1, we evaluated for both clones. If the expression was adequate, I mean, if you were having expression in leiomyoma, there should be something very, very wrong. So this was evaluated, if it was adequate, if there was any sort of background that would constitute a concern. Please see that all these stainings were optimized carefully. So, I mean, this is not wild expression, this is not research environment, this is clinical environment. And then intensity of expression. And as far as PD-L1, here's what we got. So, for both clones, we understood that the specimens in the overall were adequate for the expression. We didn't have any weird expression after these processings and evaluation. And of course, we were not expecting this. These processors are approved for use, although they are very different from each other. We have processors that have microwave, we have processors that don't have microwave, so they have different technology involved. Some are old models, some are more recent models, and we understood that in the overall they were adequate, so we could not see any relevant alterations by the human eye. In the background as well, there was no difference. There were 1 or 2 cases with background, but we could immediately understand it was a background and we could filter. Again, this is human evaluation. We were not introducing AI here at this point. And then we evaluated the intensity of the expression. In leiomyoma, we didn't have any expression, so it was good. All cases were correct, but then differences started to pop up. So in the placentas, we had differences in PD-L1 expression of both clones, and this is related with intensity. So processor A versus processor B, we had significant differences in intensity of the expression of both 22C3 and SP142. So B had a more intense, a more open expression, while A and C were very similar, and then B was again different from C. So tissue processing and having different types of processes within the same lab will impact the expression of PD-L1 in the samples. And this is, I mean, completely ignoring other variables that we may have, pre-analytics and so on. And here you may have a picture to illustrate precisely that. You can see in the 22C3 raw column, I'm sorry, you can see that A and B are very, very similar. We don't separate them, but in the C, it's more faint. So it's a different expression. And about SP143, you have something very similar. The E picture, it does have more intensity than the other, than the D and F pictures that have less intensity. So here you may see that these influence is evident, and ultimately when it is taken into the maximum extension may correspond to a false negative. If we don't have enough expression, if we don't have enough endogenicity, we may lead into a false negative unnecessarily just because of processing. And here in tonsil you may see something that was more evident in SP142. We had differences of expression that were related with processing as well. So processor C was generating tissues with a low antigenicity. So in a way, the expression of PD-L1, SP142 in the germ centers was significantly decreased. And here we are dealing with controls. How about agent D? In the overall, we dedicated ourselves to understand, oh, sorry, something went wrong here. We dedicated ourselves also to evaluating agent D if there were artifacts or not. Related with processing, I'm not talking about folds, uneven thickness of the cutting, no. Talking about decreased cell size, chromatin fading, loss of detail, and we understood that these alterations were quite frequent. They could be present in up to 50% of the specimens. And in the overall, and our daily work, we are not taking care of this. I mean, we are tolerant again, but if we pay attention, we will find these alterations. And we understood that processors were again generating different percentages of samples with artifacts. I mean, in the overall, they had no impact in the diagnosis because we could, I mean, our brain could overcome this, except with one case. In this case, you see here was a leiomyoma that was full of artifacts, and these artifacts were transversal to all processors. So we think that this was pre-analytics, very strong pre-analyticals issues. But in the overall, this was not impacting. They were there, but they were not impacting. So this leads us to question, is this going to impact AI or not? I confess that we didn't test it by our hands, but others have tested, and the influence is impressive. So what's happening in this point? We are understanding that to evaluate immunohistochemical techniques, to evaluate immunohistochemical biomarkers can be tricky, and this quantification can be completely biased when we are using AI softwares. So what are researchers doing? They are trying to do evaluations of the status of PD-L1 only based on agent D. So trying to predict PD-L1 status after agent D without having to perform the immunohistochemical testing. This group, that is a group from Israel, we participated in the study as well, and we understood that it's possible to reach areas under the curve of 0.9. So it is extraordinary the way these softwares can predict in a very accurate way the expression of PD-L1 per sample. So this is one way of overcoming all issues associated with immunohistochemical technique. Other groups are also trying to do some prediction, and here they don't even pass by PD-L1, they go directly from agent D to prediction of therapy response, for instance, for nivolumab here in this case. My issue with these types of studies is the design of the control of these instruments. They are very difficult to control as well, so we never know if they are operating properly. Ultimately, processing will interfere as well with the AI interpretation, so ultimately controls will be needed, and more studies will be needed as well, namely dedicated to the interfering of processing. Another way of trying to overcome our techniques, and here it's the ultimate overcoming agent E, it's the use of the virtual staining. So based on the fluorescence, the autofluorescence of the tissues, so without introducing any bright field stain, it is possible to collect information that is translated in a digital image that generates an artificial agent E, and nowadays it's also possible to generate artificial immunohistochemical staining, for instance, for HER2, as the one demonstrated here in this work by the group of Eidog and Oskan. So what are our messages for this work? I mean, there are many tissue processing modalities. They will carry different impact in our samples. They will carry different amounts of artifacts. They will carry different antigen exposure, and in a way it could be convenient in a short term to reach a point of standardization of processing that could be universal for pathology, in a way that we all know that the machines have a certain interference that has a range. If this is not standardized, interference can be very wide and different to control. We are very resistant to these processor-driven alterations. As humans, we can tolerate under the microscope, even under the screen, but this is not a feature of AI. So if you want to be prepared for AI, and we are certainly not prepared for AI, we need to have a more standardized lab product. And of course, this comprehends alterations in tissue processing. Tissue processing modalities interfere with the expression of immunohistochemical biomarkers, and this includes PD-L1, and may lead to false positive and false negatives, mainly false negative results. And this technology of processing needs to evolve, definitely, to more robust technology that, of course, need to be more homogeneous, as I was saying, with faster results, because this is also the added value of pathology, to have fast results, and not less important, to have clean technology. We are seeing there is a tendency to have cleaner reagent, less toxic for the operator in the lab, and this is very, very relevant. We know that the diseases, the professional diseases that emanates from exposure to toxic reagents are there, and definitely we need to protect our teams. So quality control for tissue processing is needed. It's very difficult to establish, and definitely it's something that underlies the safe morphological diagnosis that ultimately we need to observe to prevent AI bias. And this is what I had to share with you today, and I really look forward to hear your comments and questions.
Dear Professor Eloy, thank you for your excellent presentation, and also interesting and very provocative. So we have to reevaluate some of our basic steps in our labs. I start with one question following your presentation. You utilize 3 different processors, and one of them seems to have a different final result. Could you give us some more, not the company of the processor. But if there is a different time of processing, different reagents, different temperature, different modality, if there is any clue for these differences?
Well, the processors are very different from each other. What I can say to you is that some are very old models. We introduce here a very old model with a very ancient technologies, a carousel processor. And to be honest, this was a processor that was performing quite well. So old technology, it's not so automated, but it is quite robust in maintaining antigenicity. Where we feel the issues is when we start introducing very high temperature. When temperature is very high and microwave is very aggressive, we know it will accelerate processing in a certain extent. But very high temperature will obliterate the exposure of antigens. I don't know the mechanism, the way this happened, but probably there is denaturation of proteins and antigens are not exposed the same way. We know, for instance, for PD-L1, that temperatures for immunohistochemical processing and for the regular processing of immunohistochemical reaction cannot go above 65 degrees. And there are some processors that go beyond those temperatures. So in a way, what we are understanding is that probably processors that are using very high temperature are damaging the antigens that allow us to identify PD-L1 expression, and this goes towards the false negative reaction. So immediately making the tissue silent because of that. In a way, there are many variables in each one of them. They are using different reagents. Of course, these different reagents may interfere differently. We had no conditions to separate them and to mix them, even because if we are introducing different reagents in different instruments, we may kill these instruments. So it's dangerous to do it. But I mean, industry now has at hand the knowledge to try to investigate deeply this interference. Definitely, and it's up to them to improve the instruments.
So the high temperature is probably in a certain phase of the processing of tissue is damaging, but we utilize antigen retrieval that is well over 100 degrees. That is instrumental for the good result of immunohistochemistry. So we have to understand better what is happening in the tissue that is a block of tissue, or in a slide where the high temperature is a good, it is a necessity. So we have to understand better also the basic aspect of the sample preparation. Yes, this is a very critical point that we ask companies to investigate, but also as a pathologist we have to be prepared for this. And I have just another question. You mentioned that there are some antibodies that are well established as a companion diagnostic for PD-L1. But the antibodies against PD-L1 now, there are a lot of different antibodies, cheaper antibodies, and some lab utilize them. What do you think about this aspect that is also another point of variability in the testing this biomarker that is quite important?
Yeah. So giving the point where There are many variables that we definitely cannot control, and some of them I bet we don't even put in the equation because we did not remember and we are not aware of. I would recommend that people stick with the recommendations for the determinations as much as possible. So if there is the possibility to have at hand the 22C3 for determination of response to pembrolizumab therapy in non-small cell lung cancer, please use it, even if it's more expensive. I understand that precision pathology has a price. Precision pathology is not low-cost pathology. It's high cost. If we want to have an excellent product, if you go to a shop, you want to have a beautiful model of something that is extremely good, you don't pay cheap, you pay high. And here is the same for pathology. So do not go cheaper and jeopardize your results providing poor quality information to our patients and clinicians if you have the possibility to have at hand something that is more adequate and has demonstrated effectiveness on the analysis. It is, however, impossible for us to have at hand all the instruments, all the antibodies for every determination. In the majority of the situations, because funding is not illimited and we are not just working for PD-L1 determinations, we have many determinations to perform. So to have one that is the most frequent or have two that include the most frequent, for instance, I told you that 22C3 is very different from SP142. They are measuring different things. And we know that the interchangeability of them, it's not so good. So probably you should have both to cover this different range of evaluation. But then SP263, it's quite similar and it's as much as robust as 22C3. So to have one of these with SP143 could be a good balance to have in the lab. These two could be a good balance to cover the range of variability of expression. To have non-recommended antibodies, I mean, it's possible to have satisfactory results. It is a dangerous situation when you are dealing with borderline tissue conditions, tissues that were hyperfixated or less fixated, just in a small extent, could suffer from the introduction of these antibodies, the much they look very reliable. So yeah, to understand this in our minds could be good. Precision has a price, and probably to go for cheaper things, it's not the best way to go.
Thank you very much for your presentation, and I hope that the message will take home your message. Thank you very much. Bye.
Thank you.